Let's look at the effect of pH, temperature, and ultraviolet light on bacterial growth for this lab. First of all, pH. pH dictates bacterial growth because you put it in an environment that the bacteria doesn't like. It does alter the protein structure. It can damage the cell membranes. It can interfere with energy production. Because if you remember, bacteria do not have electron transport chains, and they must use they don't have mitochondria. So they got to use that cell membrane to make energy. And the bacteria have to maintain a internal pH to survive. And if it gets in an extreme environment, it disrupts these cell functions. It will slow their growth or cause cell death. So how it affects it, hopefully you guys have learned in bio that, when you denature a protein, when you expose it to acid or heat, we'll talk about that. Basically, you're breaking down that 3D structure of the protein, and we call it the quaternary structure. It's how the protein folds. And so altering that pH, that alters the electrical charges on those amino acids and the hydrogen bonds that hold their shape. And so these proteins that have to be folded correctly unfold and lose their function, and especially those proteins that are enzymes. Okay. If you have a highly acidic or basic condition, it can degrade the lipid bilayer of the membrane, causing it to leak or break down. And bacteria rely on this gradient of protons, the hydrogen ions, again, to make energy. This is how they make energy rather than and generate ATP rather than going through how they go through glycolysis, if you will, and instead of going through the electron transport chains. So if you have a very drastic change in pH, it destroys this gradient. So we classify the pH into 3 categories. You'll see this commonly: the neutral guys, the acid guys, and the alkali guys. So these guys love the neutral pHs, typically close to 7, within a couple of pHs. Most of our pathogens, not all, but most of our pathogens and a lot of the bacteria that are common will fall into this category. Now, there are those guys that like an acidic environment. So pH of 5.5, I'll show you some of them. And then there are those that like an alkaline environment, and they require that to grow. So this is how the classification works. We call them, okay? And then we have the optimum pH. That is the most favorable pH for that organism to grow. The lowest pH it can tolerate is its minimum growth pH, and the highest pH that it can tolerate is its maximum pH. And these can be a pretty wide range depending upon the organism. And I gave you the example of Salmonella. Its optimum pH is 7 to 7.5, right around ours, but the minimum pH is closer to 4. So that's a pretty big range for Salmonella. When you get into these neutrophils, these bacteria optimally grow within, again, 1 to 2 pH units of 7. Your most familiar bacteria like E. coli, staph, Salmonella, they're neutrophils. They don't fare well in the acidic pH of the stomach. Okay. There are pathogenic strains, however, that are much more resistant to the stomach acid and can survive. Fungi like slightly acidic values, so each one is kind of a little different, and the stomach acid is protective. We'll talk about that when we get to diseases of the digestive system. I'm just trying to give you an overview. One of the examples we'll talk about a little later too, that's unique. Some of these organisms can adapt to this stomach acid and be able to survive. So Helicobacter pylori, which causes our peptic ulcers, used to be we thought it was spicy food or stress or whatever, and we'd be giving antacids and all that sort of thing. Well, the real cause is this bacterium called Helicobacter pylori. And although it is a neutrophile, it can survive because it creates a microenvironment around itself in which the pH is nearly neutral. And what it does is it has an enzyme called urease, and it produces it in large amounts, and that breaks down urea, which is a breakdown product of protein, to form ammonium and carbon dioxide. And the ammonium ion raises the pH, because ammonium is an alkali, raises that pH of that immediate environment around that bacterium. Okay. Then we have the guys that like it basic. So they grow best at these higher pHs. A good example of this is Vibrio cholerae. This causes cholera. It grows best at a slightly basic pH, which, FYI, is seawater. And this is why seawater can be a reservoir for Vibrio, because it will do very well. It can survive at pHs of 11, but is inactivated by the acid of the stomach. And again, we'll talk about these guys when we start talking about diseases. Temperature is very important. There are several environmental factors that go into these guys. The temperature is most commonly linked to enzymatic activity and structure because of the enzymes being proteins. And again, that quaternary structure, it has to be right. When you heat these up, you denature these proteins. You may or may not fully do it. We'll talk about it. Or you can make it so if you bring it down too cold, it's too stiff really to work in there. So again, we have our cold guys, our middle guys, and our hot guys. Okay. The effect of the temperature, again, the catalysts that speed up enzymatic reactions. These are proteins, these are enzymes. They have a lock and key system. They have to be able to bind to their substrate at the active site to work. And if that folding isn't correct, you don't have a working enzyme. So we have an optimal temperature at which these enzymes function. And again, if they are exposed to a temperature higher than optimal, the structure of that enzyme may be altered permanently or may be able to go back. So if the structure of the enzyme is altered, it can't attach to the substrate. It's a lock and key system. Permanent alteration of the enzyme with it not functioning anymore is called denaturation. You can have thermolabile, which these organisms are able to withstand slight increases of temperature above their optimum without denaturing. Or you can have thermostable, which can tolerate fairly moderate temperatures above their optimal temperature without permanent damage. I mean, there are plenty of proteins that do that. I like to do this when I talk about denaturation, but think about an egg. The albumin of an egg is a protein. You put that egg in a hot frying pan, and that egg white turns white. And you can't ever get it back to its original consistency. So that has been denatured. Whereas if you take milk, it's much more thermostable, and you can heat the milk up and you can cool it back down and you still got milk. All right, so this is kind of like that kind of theory. Then you have the guys that like it cold. We have to worry about these because a lot of these guys will grow very well at refrigerator temperature. And some of them can, you know, grow in refrigerators, depending upon what it is. But a lot of these guys like refrigerator temperatures, and this is why you can, see growth in the refrigerator. You get mold growing and all kinds of nasty things can grow because it's not enough to kill them. All right. The mesophiles like you can see just below 20 degrees. So just, a little bit below room temperature, a little bit above our normal temperature. So they grow very well within these temperatures, and which many of our foods are held for consumption. So you have to be very careful with this food handling because these guys can live at these temperatures. Okay. Because of the importance, we do classify them, we subdivide them. The room temperature loving guys who grow 20 degrees to 35 degrees Celsius, these are plant pathogens. They also help with the decomposition of plants and animal bodies. And then your body temperature mesophils, these are found on the warm-blooded animals. They are mutualistic. These are the guys that are our normal flora or our pathogens. They prefer our body temperature, and they do very well there. Then you have the thermophiles. These, they're very thermostable enzymes, and they do well in these hot environments. One in particular that I want to mention, and I'll talk about this also when we get to respiratory diseases, is Legionella pneumophilia. Pneumophilia meaning lung-loving. Legionella, this goes back in the history into the 1970s when the American Legion was having a convention in Philadelphia, Pennsylvania. And these veterans, a lot of them got sick. And some of them died, and nobody could figure out what it was. So they finally, and we'll talk about how, but they basically found out that there was an organism there. And this organism, Legionella, likes warm water, likes hot water heaters, likes hot tubs. I have seen several cases of Legionella pneumophilia from hot tubs. Particularly, we had an outbreak in the Keys of Florida, a contaminated spa. So if you like to go and visit spas, or you're working and draining your hot water heaters, be careful. Make sure, but particularly the spas, because you're sitting in that water and breathing the fumes off the water. Make sure it's been treated. Make sure that hopefully you don't get Legionella. You can also see these guys in warm springs and geysers like Old Faithful. They do very, very, very well. So here's your temperature range of bacteria. Okay. And you can watch this video on the effect of the temperature on the microbial growth. I found a good YouTube video on this and goes through, actually takes you through the experiment so you can see this stuff. One thing I wanted to notice is when I actually do the lab, I have several different bacteria and we look at them. But what's really unique is this Serratia marcescens. And Serratia is pretty clear at 37 body temperature, but you put it at room temperature, it is this beautiful pink color. So hopefully you'll get to see this in the video. So some of these organisms do change colors at different temperatures and allows us to see them. All right, the effect of radiation on microbial growth. Now we're going to switch topics here. And you have the visible light, Roy G. Biv, the longer wavelengths, which is infrared, radio, microwaves, and the shorter wavelengths, which are higher energy and particularly ultraviolet, because we can use ultraviolet to get rid of these guys. Okay. When we try and kill these organisms in a healthcare facility, situation, you don't want, you don't want the patient is usually somewhat immunocompromised, and you don't want them exposed. So you want to control the growth of these organisms in materials and on materials. Some of these things you can use heat, and we'll talk about this, autoclaving, but some of these items can't be autoclaved. Okay, you can't autoclave a plastic instrument. You can't autoclave, an endoscope that has pieces and stuff like that. If it's all metal, it's not a problem. But some of these things you can't. You can wipe down a surgical table, but it's going to be cleaner if you have your ultraviolet light and let it go on there. So it is most effective at 260 nanometers. This is the specific wavelength where DNA does maximally absorb the ultraviolet light. The one thing you do have to worry about with ultraviolet light is it cannot penetrate glass or plastic or even paper, any solid or liquid that may be in between, okay? You can do like a surface of it of clear liquid, but nothing else. So you can see the different kinds of ultraviolet light. And again, if you go back here, it's 260 nanometers, which puts us right in this UVC range. These are very dangerous. Ultraviolet light can damage your eyes in seconds, so you don't want to expose your eyes to these. All right, now what does UV light do? It causes what's called thymine dimers. And what does that mean? When you look at a strand of DNA, you usually have this strand and this strand, and these guys are bonding. So the thymine is bonding to the adenine to the thymine, the cytosine to the guanine, and so forth. When you get exposed to ultraviolet light, what happens is instead of binding across like you normally would occur, the thymines bind together in a double bond, and that's why it's called thymine dimers, okay? Bacteria have a repair system that can go back and repair the damaged bacteria, but it depends on the exposure time to this light. If it's real quick, they can repair. If it's longer, it will kill them. If you have an endospore, it's going to take longer to kill these guys. So what I do in this lab, when I do it in a lab, I have my students compare an endospore forming bacillus and a non-endospore forming cocci, like staph. So, and there's a big, huge difference. The endospores are formed by some of these bacteria that have the ability to enter that state of suspended animation. We talked about endospores, and basically it will move its DNA into this endospore when conditions are not favorable for that bacterium to survive. So and that's going to protect it. It's going to make it harder to kill using this UV light. So what we generally do in this lab is dip a sterile swab into the culture. Again, you got two cultures. Streak the swab straight down the middle and just start at the top and cover your whole plate. Turn it 45 degrees, 90 degrees. Just keep turning it around, bottom line. I rotate it 90 degrees, streak it top to front, and then 45 degrees. And then I swab the total edge of the auger. You can watch this. This is a video of somebody doing this. But the bottom line is you cover the entire plate with the bacteria. And so you label the plate, you put a line down the middle, and you place it in the UV hood and remove the lid, because if you do not take the lid off, remember, anything between the UV light and whatever you're trying to sterilize, it will stop the UV light. So you must remove the lid of the agar plate to expose it, because if the lid is left on, everything will grow in that plate. So you remove the lid and you cover the covered half with an index card. That is to show you, bottom line is that UV light cannot pass through that piece of paper, right? And then you expose the plate to specific times that I would set. And then when it's done, you place the lid on the top and you place it in the incubator. This is the procedure that I would always do with my students. And it should say plates and not plats. All right, and this is what you get. Okay, so this obviously is the side that had the card, the index card, because you have plenty of growth and that UV light was not able to make it through it. This side did not, and it was exposed to a time enough to kill the organism. But you can see that confluent growth. It is all over this plate, okay? What we usually see, what I want you really to gain out of this is what we normally, since we can't do it live, what I'm going to do is give you the results of, many, many, many micro labs that I've done with my students. And staph epi would be, I would have them expose them at 30 seconds, a minute and a half, and 2 minutes. And again, staph is not endospore forming. So staph will all be dead usually by about a minute and a half. It doesn't take very long. I use a bacillus species that is endospore forming, and I have them expose it for a minute, 2 minutes, 3 minutes, 4 minutes, 5 minutes, 7 minutes, 10 minutes, sometimes going as high as 15, just if we've got the time in the lab. Generally, what we see with the bacillus is that it will be completely dead in 7 to 10 minutes. So it takes a lot longer. So this is why, as if we're trying to sterilize something like a surgical table or something like that, when you really need to, turn the light on, leave the room, flip the light on, and let it sit there for usually at least a half an hour, and then everything will be dead. Okay. So hopefully this explains to you some of these things, and we'll see you next time.